Hi everyone, and welcome to our Daily Booster number 7 in our biology series. What we're going to be doing today is carrying on with photosynthesis, but we're going to be looking at what we refer to as limiting factors. What we're talking about here then is when we think about photosynthesis, then there are certain factors that will affect the rate of it. And this is what we refer to as the limiting factors. There are 3 we need to know about: light intensity, carbon dioxide concentration, and temperature. One little point to note here is because we do know that 10% of the marks on the biology paper will be maths based, they could throw in a question asking you to calculate the rate of a photosynthesis experiment. If they do, all you need to do is 1 divided by the time it took for that measurement to occur, and that gives you a measurement for rate. What we'll do then is we'll take each of those factors in turn and have a look at the graph that we need to recognise and also be able to recreate, should we be asked to do a sketch, to explain what's actually happening. So the first one, light intensity. The general pattern is that if we increase the light intensity, then we have a faster rate of photosynthesis. However, the graph will not just keep increasing forever. If you look at the little picture down here, we can see it levels off. So the reason that our graph actually levels off is because at the point that it becomes flat, so horizontal, then light intensity is no longer the limiting factor. Carbon dioxide concentration or temperature is now limiting this reaction. So increasing light intensity will do absolutely nothing to increase the rate of photosynthesis beyond that point. For those of you doing higher tier, you will need to know about the inverse square law as far as this is concerned. And the inverse square law just states that when you double the distance from a light source, the light intensity decreases by a factor of 4. If we want to represent that in a mathematical way, then you can see just underneath, relative light intensity is 1 divided by the distance from the light source squared. So that's why if we double the distance, the light intensity decreases by a factor of 4, because obviously double means 2, so 2 squared is 4, therefore 1 divided by 4. What we're doing is decreasing by a factor of 4 there. So you can see the simple way of writing it, just 1 over d squared. Factor number 2, the concentration of carbon dioxide then. When we're thinking about the concentration of carbon dioxide, our general pattern is that the higher the concentration, the faster the rate of photosynthesis. Now again, we see the exact same shape graph that we saw for light intensity, increasing initially and then levelling off. And the point that we level off, that is of course because carbon dioxide is no longer the limiting factor. So something else is now limiting the rate of photosynthesis. The third factor, temperature, is a little bit different. Hopefully we know that photosynthesis is an enzyme-controlled reaction. By that we mean for photosynthesis to take place, it involves enzymes. We have already in one of our earlier boosters had a look at how temperature affects enzyme activity. And the nice thing is, it's exactly the same here, except we're just talking about a specific example of photosynthesis. So if we look at the graph, it should be incredibly familiar to us from the enzymes one. So initially we can see increasing the temperature increases the rate of photosynthesis. The reason being that as we increase the temperature, the particles gain kinetic energy, they're moving faster, and therefore we have more frequent collisions. So more reactions take place in the same time. We do then get to our optimum, which is the highest point on our little graph there. And the optimum is the temperature at which photosynthesis is happening at its fastest rate. If we keep heating it up and go well beyond the optimum, you can see that it's then very rapidly drops off. Now, the reason that we get this very steep decline in the rate of photosynthesis when we go to these much higher temperatures is that we are denaturing the enzymes. So think back to the work on enzymes that we've done. Enzymes have got this very specific shape of their active site that's complementary to the substrate. If we denature the enzyme, what we're doing is we're changing the shape of that active site so the substrate no longer fits. As a result, the reaction can no longer occur. So this is what we're seeing in the case of photosynthesis. Increasing temperature initially leads to an increase in the rate of photosynthesis because the particles are more likely to collide because they're moving faster. We have our optimum where it's working at its fastest rate. And then as we go beyond the optimum, as we go to those higher temperatures, the enzymes denature and therefore we get a steep drop off. Do remember that key phrasing here, that when we're investigating any limiting factor for photosynthesis, there will come a time when the graph levels off. So when we're looking at light intensity, the most common one, you won't be able to just keep increasing light intensity and get an ever increasing rate of photosynthesis, because you will hit a point eventually where your graph is going to plateau, level off. And that's because there is a different limiting factor involved. So just bear in mind that key point about limiting factors. Of course, increasing one factor is going to increase the rate initially, but then something else will take over. Time now to head on over to have a go at the quiz to see how well you've done, how well you've understood today's work. So the link is again in the description for those of you that don't really fancy typing that out. And of course, don't forget to join us again tomorrow for our next Daily Booster.